This video is part 2 of chapter 8, and we're going to complete our discussion of chemical reactions in the cell and metabolism and stuff like that. Now, so we've mentioned there's a variety of different types of reactions that occur inside a cell, and we call these metabolic reactions or metabolism. Well, as we've mentioned before, each step in a metabolic pathway must be catalyzed by an enzyme. So we're going to be talking about enzymes here for a little while. An enzyme is defined as a catalyst. And a catalyst is any chemical agent that speeds up a reaction without being used up in the reaction. And that's a really important characteristic. So since it's not used up in a reaction, a catalyst can activate or facilitate multiple reactions. Okay, so it can be used over and over again. An enzyme is what we would call a biocatalyst or a catalytic protein. And enzymes facilitate all kinds of different reactions inside of a cell. Now, what enzymes do is they overcome the energy barrier of reactions. Recall from the last lecture that we talked about either the need for energy or an output of energy in particular reactions. Well, the initial energy needed to start any chemical reaction is called the activation energy. In many cases, the activation energy is supplied as free energy in a system or in an environment. But in other cases, there needs to be an input of energy to make the reaction go. If we look at a diagram of this, let's say we've got two reactants here, and they require a certain amount of activation energy to get the reaction started. Well, once they get to that activation energy, or once enough energy is supplied, then they can proceed and complete the reaction. Now, as I said earlier, sometimes this activation energy is just based on free energy available in the system, and all the reactants need to do is encounter each other. But in many cases, that's not the situation. So what enzymes do is they catalyze reactions by lowering this energy of activation. Now, they don't affect free energy at all, but what they do is they lower the activation energy barrier and make reactions go faster or facilitate them. Here's what that would look like on a similar graph. So if we have a reaction that starts with reactants at this level, needs this amount of activation energy to go in order to proceed to the products, well, an enzyme might come along and lower this activation energy. So notice this is considerably lower than the activation energy without the enzyme. So what that means is the enzyme makes the reaction go more quickly, or it just facilitates it going or proceeding without any excess energy input. Now, enzymes function in very specific ways, and we're going to go through that in the next few slides. The reactant that an enzyme acts on is called the substrate. So any molecule that an enzyme does its job on is a substrate. What happens first is the two molecules bind, forming what we call an enzyme-substrate complex. And then the reaction catalyzed by each enzyme is very specific. So each enzyme catalyzes a reaction with a specific substrate. So there's very few enzymes that will react with more than one different substrate. The spot on the enzyme in which the substrate is going to bind is called the active site. It's sort of like a lock and a key. So the lock is the enzyme's active site and the key is the substrate. When they bind together, we call this an induced fit because they fit so closely together. And in many cases, they form chemical bonds. Most of the time, it's hydrogen bonding. In other cases, they may form covalent bonds between the two molecules. Now, what the enzyme's active site does is it lowers the energy of activation, and it does this in a number of ways. One way is that it orients the substrate correctly into the enzyme. The active site orients it into the enzyme so that the bonds can be altered, or that the substrate can be altered. This causes a straining of the substrate bonds. It also provides a favorable microenvironment in the enzyme to alter the substrate. And as I mentioned, in some cases, the substrate is covalently bonded to the active site in order to alter the substrate. Here's what that looks like. And this is a 5-step process. This is the this is the catalytic cycle of an enzyme. First of all, the substrate enters the active site. In this case, there's two substrates. That's common. The substrates are held in the active site by weak interactions such as hydrogen bonding. The substrates are converted to the products, and the products are released. Then notice that the enzyme goes back to its original structure in order to facilitate the same reaction again. As we mentioned earlier, that's a really, really important characteristic of catalysts or enzymes, that they don't change structure throughout the reaction, and they can go and facilitate the same reaction numerous times. Now, local conditions affect enzyme activity significantly. And in the human body, for example, temperature and pH have a lot to do with how an enzyme can function and if an enzyme can function. And then there are also chemicals that can specifically influence the enzyme as well. Each enzyme has an optimal temperature in which it can function, meaning there is a range of temperatures in which a particular enzyme can function at its highest efficiency. The same goes for pH. And optimal conditions favor the most active shape for the enzyme molecule. So in some cases, temperature and pH can alter the shape of an enzyme and therefore alter its effectiveness. If we look at a couple different graphs here are two different enzymes that operate at very different temperatures. So the optimal temperature for a typical human enzyme is going to be 37 degrees. So if we see rate of reaction on the y-axis, the highest rate of reaction is at 37 degrees. Well, if we look at a heat-tolerant bacteria that lives in hot springs, its most effective temperature is at around 77 degrees Celsius. Well, at that temperature, a human enzyme is not going to function. By the same token, at lower temperatures, a heat-tolerant bacteria will not have any enzyme function because they are adapted for the higher temperature. If we look at pH, an enzyme that functions in the stomach of a human is going to function optimally at pH 2 because that's the pH inside the stomach. However, an enzyme that operates in the intestine of a human is going to operate and function most effectively at pH 8 because that's the pH of the intestine. Now, besides chemical environment and temperature affecting enzyme function, enzyme function is also regulated very closely inside of a cell. Because if the cell's metabolic pathways were not regulated, there would just be chemical chaos, as the slide states. So what the cell can do is it can turn enzymes on and off, or it can turn genes on and off, and both of these can affect the activity of enzymes. One type of regulation is what's called allosteric regulation, and this may inhibit or stimulate an enzyme's activity. This usually occurs when a regulatory molecule binds to an enzyme at a site other than the active site, and it can change the structure of the enzyme and therefore change its function. Most enzymes have active and inactive forms, so consequently the allosteric activation and inhibition is either going to turn an enzyme off or it's going to turn an enzyme on. So allosteric activation will turn it on, allosteric inhibition will turn the enzyme off. One way that this can occur is through what we call feedback inhibition, and this is a type of negative feedback which we've talked about before. This is where the end product of a metabolic pathway shuts down the pathway. What this does is it prevents the cell from wasting its resources by synthesizing more of a product than it actually needs. Cells function really on an as-needed basis, so if a molecule is needed, it's going to be produced. If the quantity of the production exceeds the need, then the production is going to stop. Here's how that might work. We've got an enzyme here that's going to activate a particular pathway, and here is the end product. Well, as that end product builds up, it's going to bind to the enzyme and alter the active site. If we notice here, here's what the active site would look like when the enzyme is functional. Here's what it looks like when the enzyme is non-functional. So this allosteric inhibitor binds to the enzyme and changes the active site so that the substrate is no longer going to fit. Well, if this molecule decreases in amount, it's going to be stripped off of the enzyme and it's going to return the enzyme's active site to its normal condition and therefore allowing the pathway to proceed.